Hi everyone, so today we're going to be talking about chapter 4, which is carbon and the molecular diversity of life. Again, this is a really quick chapter. The main function is just the different forms of carbon and then functional groups, so we should get through this pretty quickly. So carbon, as some of you may already know, is the backbone of life. There's a whole study of chemistry related to it called organic chemistry. So living organisms' chemical makeup is based on carbon. All living things have carbon in them. So it enters the biosphere through the plants. They're able to convert carbon dioxide into a usable form for us. And as you know, is glucose originally and then converted into other forms. And then we eat those molecules. So we cannot use carbon dioxide. We exhale it, then the plants use it to create food. And then it's the main components of macromolecules. Within chapter 5, you'll be learning all about carbohydrates and lipids, proteins, and nucleic acids. All of those have carbon as the backbone of them. Okay, so like I said, there's a whole branch of chemistry studied that is devoted to the study of carbon, and that's called organic chemistry. And it's because it's so versatile. It has a lot of unique characteristics that other things do not. And one is that it has 4 valence electrons. And so since it has 4 valence electrons, that means it can form 4 covalent bonds. It's called tetravalent. It has a tetrahedral shape, as you can see here. This is 1 carbon with 4 hydrogens. So that's the simplest organic compound, which is methane. It can also bond to itself in order to form long chains or rings. Okay, and then one other thing that you need to keep in mind is that carbon dioxide, although it contains carbon as CO2, is actually an inorganic compound. And it's the source of carbon for the organic compounds. So when we go from carbon dioxide to an organic compound, it's called carbon fixation, kind of like we talked about nitrogen fixation with our last unit, where we're converting unusable nitrogen to a usable form. The same thing is happening here, but we're converting carbon dioxide into an organic form, usually in the form of sugar. Okay, so hydrocarbons are the first thing we'll talk about, and those are compounds that have only hydrogen and carbon. When we looked at on the last slide was an example of methane, which is the simplest example, and here's another picture of methane. Petroleum is a huge hydrocarbon. All of our fossil fuels in the world, hydrocarbons by themselves are nonpolar, so remember that means that they have an even sharing of electrons, so they're not attracted to water. They will not dissolve in water. A huge benefit of hydrocarbons is that they store large amounts of energy, which is why we burn them for our energy, for our fuels. Something else that's really important in biology with carbon are that they are isomers. And isomers are compounds that have the same molecular formula, but they have different structures. So they have the same formula, like in this example, you have 4 carbons and you have 8 hydrogens, but they're different. This one, butane, is just a straight line. Isobutane has 3 in a straight line and then a carbon coming off of the second one. So since they have a different structure, that means that they also have a different function, because structure dictates function. So since they have a different structure, it has a different function because they will have different properties. So you can see that there's all of these different examples, and we'll talk about those a little bit more. All right, so one type of isomer is a structural isomer, which differs in the covalent arrangement of the atoms, which is like the one we just talked about with butane. Here's another example with pentane. So a straight line of 5 carbons, or 3 carbons in a branch, or it could have been 4 carbons with a branch. It doesn't matter. It's a different structure. The second type is geometric, which differs in the arrangement about a double bond. So it depends if these X's are things other than hydrogens, and so they're in different locations of the double bond. So on this side, they're both on the same side, and this one you have one on the top and one on the bottom. So that would give it a different structure, therefore a different function. And then enantiomers are mirror images of each other. So the middle carbon is asymmetric, and it's considered to be what's left and right-handed, and so if you put your hands together, they have the same chemical formula, but everything is flipped, like mirror images. Okay, enantiomers are really important in the pharmaceutical industry because you can have different medications if you have the wrong form. So you have the thalidomide, which is a mixture of 2 enantiomers. One is an effective drug, and it reduces morning sickness. But the other form caused birth defects, and they didn't really know about that until after they had been prescribing this medication. And so obviously you don't want that to happen because the cell can tell between the two isomers. It causes these differences. And then ibuprofen and albuterol are also a mixture of enantiomers. So ibuprofen, is a pain reliever, and that's sold as a mixture of the two. And then albuterol, which people with asthma take to relax their bronchial muscles. So if they have an inhaler, it's usually albuterol, it's in it. The R-albuterol, which is the one form, is synthesized and sold. That's what you're taking if you have it. If you have asthma, you have to take it. And then S-albuterol counteracts that form. So obviously you don't want to be taking that. So different effects, even though the difference is only where they are in the carbon, it still causes huge differences. So here you can see the differences. So S-ibuprofen and R-ibuprofen is just the location of these probably oxygen molecules. And then albuterol with asthma, you can see that the location is flip-flopped. Okay? All right. The next thing we need to talk about then are functional groups, which are organic molecule properties due to the arrangement of carbon along the components. So functional groups are groups of atoms that are attached to the skeleton. They're taking the place of the hydrogen, and then it's kind of acting as a hydrogen. It's acting as a group together. They have chemical properties based on that arrangement. And then the number of groups and the arrangement of those gives each molecule a different property, and it will react differently. So these are the ones that you need to know. We have hydroxyl, which is OH, and that's called an alcohol. Anything with an OH group at the end is an alcohol. These are hydrophilic, so they have polar covalent bonds and they attract water. So here's some more some more information about it. Then we have carbonyl. There's two types of carbonyls. Carbonyl is a carbon with double bonded oxygen to it. They are also hydrophilic. And you have two types based on where that double bonded oxygen is located. You have a ketone, and that's if it's in the middle of the compound. So like in this example you see here, you have 3 carbons. This would be the simplest ketone because it's in the middle of your compound. Aldehydes, it's at the end. So again, there's the difference is where it's located, and it'll give it different properties. Okay. Carboxyl is a C double bonded O to an OH group. So here's our structure. C double bonded O and an OH. These are also called carboxylic acids. So they donate this hydrogen that's here. They donate that, and as we talked about in our presentations, they talked about how acids are an increase in the amount of hydrogen ions. So this is increasing that. They are also hydrophilic, and you have your electronegative oxygen pulling your electrons. Remember, oxygen is very electronegative. Carbon is not. The next one is an amino, which is an NH2, and that's a base, also hydrophilic. So it's a hydrogen acceptor, so it's the opposite of our carboxyl group. Instead of donating hydrogens, it's going to absorb hydrogens. And NH3 plus is our most common form. Okay, so it acts as a base as opposed to an acid because it's absorbing those hydrogens. Sulfhydryl acts very similarly to hydroxyl. It's because sulfur and oxygen are in the same family. So it's SH as opposed to OH, also hydrophilic. It's really important in protein structure. It helps to form some cross bridges and some hydrogen bonds that keeps your protein together, which gives it its shape. We'll talk more about those bonds later. But you can see that it's attached to a lot of proteins. Phosphate is PO4, and it is super important in energy. We'll talk about ATP. ATP is adenosine triphosphate, so it has 3 phosphate groups on the end of it, and since it has a -3 charge, you have a lot of energy in between those bonds that are being shared there. So as you break those bonds, you can release a lot of energy. So phosphate is extremely important with energy, and that's mainly because of the -3 charge that's associated with it. Methyl groups is CH3. And these are nonpolar and hydrophobic. It's our only nonpolar one. They're hydrophobic because carbon and hydrogen have a very similar electronegativity. They're nonpolar. But with oxygen and carbon or nitrogen and carbon, they have very different electronegativity, so you have a big difference, and that makes it polar. So this methyl group is CH3. It's nonpolar. It's hydrophobic. Methylation can change DNA and change the functioning of DNA. It prevents some of the DNA from being transcribed and made into proteins. So that's some of the functions of that. Okay, and that's it for this chapter. If you have any questions, let me know. Otherwise, we will discuss this a little bit in class, and you have a quiz on Wednesday on those functional groups, so make sure that you have those memorized by then. All right, have a